So let's use some fruit fly examples here. So let's say we have, well, we've been using Apterus. Let's say we've got Apterus and sepia with an eye color, and let's say black body. We cross that to wild type. And let's say we know that all these guys are recessive and all these guys are dominant. Then we'll get something that's heterozygous for all three genes. And then let's say we cross those together in our F1. So what kind of cross is that? That's a trihybrid cross. So if we did this, we can calculate all different things that we would get out of it, and we'd get several different possible phenotypes. I don't know that I want to give you all of them, so let's just do a couple of examples. Let's say how many would be Apterus, sepia, and black. That'll be one example. Apterus and black, and then let's say black and sepia. And what that means, so we're not actually, when I say something's black and sepia, that means it's wild type for the other one, for Apterus. So that's wild type for Apterus, black and sepia. But when we describe flies, we only mention what the mutant phenotypes are, and then we assume everything else is wild type. So this is wild type for Apterus, so that means we're looking for a genotype like this, and then it's black and sepia. So what I do is consider each cross independently. So there's independent assortment going on. You kind of have to know that. So assuming that independent assortment is going on, and we're doing a trihybrid cross, we know there's a 3/4 probability of getting wild type for Apterus, a 1/4 probability for getting black, and a 1/4 probability for getting sepia, because those are all typical Mendelian crosses. 3 times 1 times 1 is 3. 4 times 4 times 4 is 64. Apterus and black, so Apterus, black, wild type for sepia, 1/4, 1/4, 3/4. So we had 3/64 for that. Apterus, sepia, and black is 1/4 times 1/4 times 1/4, because Apterus, sepia, black. So that's 1/64. Let's say wild type. Wild type for all three of them is going to be 3/4 times 3/4 times 3/4 is 27/64. So we just analyze each gene independently, and then we can calculate the overall probabilities. You could do a million genes. I wouldn't want to do a million genes, but you can. You might need a computer. So let's sort of generalize it, and we'll mix it up a little. Whoops. Can't have B twice. Let's say 2 little c's now, and then 2 little d's. Cross that to little a, little b, big C, big D, little d. And then we say, what's the probability of getting the dominant A phenotype, the recessive B phenotype, the dominant C phenotype, and let's say the dominant D phenotype. Well, the probability of getting A, we analyze this cross. Big A times little a. The probability of getting this big A phenotype is 1/2. we analyze this cross by itself, the probability of getting little b phenotype is also 1/2, right? Because if you got big B times little b, so I don't mind putting squares for just one gene at a time. Oops, it doesn't go there. Got little b. So we get one big B phenotype to one little b phenotype. So that's 1/2. C, so these are all test crosses in this case. Big C, little cross a little c, that's 1/2. Another test cross for this guy, that's 1/2. 1 times 1 times 1 times 1 is 1. 2 times 2 times 2 times 2 is 16. So you can do it for any combination. And you can do it for any different kinds of alleles. So these probabilities could be anything. You could do another cross where that's 2/3, this is 5/6, this is 1/12. I mean, those aren't really common, but let's just pretend that those things actually exist. You just use whatever probabilities you get for each individual cross and multiply them together to get your overall probability. But we probably won't see those kinds of numbers. We'll see 2/3. 2/3 is common. Alright, so that's that. Oh wait, speaking of 2/3. Alright, so now we have some lethal alleles and some various things. So we'll use an example. This is an example you might see on Virtual Fly Lab. We'll use curly. So if you take two curly flies and cross them together, you always get 2/3 curly and 1/3 wild type. Always. You never cross 2/3 curly, or you never cross curly and get all curly. If you cross curly to wild type, and we'll say homozygous wild type because we know if we just grab a random wild type fly, it's going to be homozygous wild type for this particular gene. You'll get 1/2 curly and 1/2 wild type. So what's going on here? Well, this, if we know this is homozygous already, a 1-to-1 ratio, that's typical of this kind of a cross. And we're going to make, so in this case, we know that that wild type's homozygous. And then we know we get a 1-to-1 ratio, which tells us that curly has to be heterozygous, because in order to get curly and wild type out, we'd have to be heterozygous. If this curly was big C, then we'd get all curly on the way out, or on the way out, on our, whatever you call it, our outcome. Alright, but we don't get 1-to-1. So what's going on here with the 2-to-1? Well, if curly has to be heterozygous, and we take this guy and say both those guys are heterozygous, then we're going to get a 1-to-2-to-1 big C, big C, big C, little c, little c, little c ratio. But what we're seeing is a 2-to-1 ratio. So what happens is we get that 2, and then we get this 1, and those guys die. So that's because big C is a lethal allele. And so it's lethal when homozygous. And that's typical of lethal alleles. They always give you a 2-to-1 ratio. Incomplete dominance, or codominance, are very similar. So let's say we take a, alright, so that's a flower. It doesn't look much like a flower. That's another flower. But we're taking a white flower, crossing it to a red flower, and I don't know if I can get pink. Let's see. That's pink. Kind of like that thing. Alright, so what does that mean? So we're taking white times red, we get pink. Cross two of these guys together, and we get 1 to 2 to 1, red, white, sorry, red, pink, white. So that tells us that this, so 1 to 2 to 1, that means it's a monohybrid cross. That means it has this form. So that's just whenever we see 1 to 2 to 1, we know it's a monohybrid cross. A monohybrid cross means that these guys are heterozygous, because that's what a monohybrid cross is. Well, if those guys are heterozygous, then these guys have to be each homozygous to give us this heterozygous. But we can't use a big A and little a because neither one of these things is dominant. If white was dominant, then all the F1s would be white. If red was dominant, then all the F1s would be red. But what we're getting, even though it doesn't really look like with this shade of pink, is we're getting an intermediate phenotype. So that means we have to have different allele designations, because we can't. So a capital letter implies that something is dominant, but this isn't dominant; it's incompletely dominant. So what we do is we use the color. In this case, the C allele for color, or the C letter for color, and then put a W on top as a superscript, and that means white. And a C, you could make it R, is red. So it makes this guy C R C R, and this guy C W C W, and this guy C R C W, heterozygous. And then C R C R C R C W C W C W. So it's just a slightly different allele designation. We would say that this is the locus, or the gene, and then this is the red version of that gene, or the allele. To relate that to chromosomes, just to go back and relate this to chromosomes one more time. If we have a chromosome, this is the locus. So we could say this is the C locus. So that's where the C gene lives. If we've got homologous chromosomes, this also has a C gene. But this is the R allele, and this is the W allele. So once again, these are homologous chromosomes, locus, and allele. So this designation and this designation are the alleles. They're different versions of the C gene that lives at the C locus. Alright, so let's do another cross with that. So let's say we're going to do a tall, we'll do a tall red plant cross to a short white plant, and we get all tall and pink. So that tells us that tall is dominant, and from before, we're using the same alleles from before, we get this. So this guy is heterozygous, and this guy is heterozygous. What do we get if we cross two of these together? if we have independent assortment. We always have to know whether or not we've got independent assortment going on. And so in this case, we do. Once we get to chapter 7, we'll start to do something other than independent assortment, but for now we're doing independent assortment. Well, let's see. Analyze each gene independently. So we've got 3/4 tall. We've got 1/4 short. Right? Because that's just a regular monohybrid cross. For the other guy, we got 1/4 red, 2/4 pink, because it's incomplete dominance, and 1/4 white. So 1 to 2 to 1 ratio. Just thinking about this cross by itself. So what probabilities do we expect altogether? We can get all these different combinations. We can get tall red, tall pink, tall white, short, 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 red, pink, white. So red, pink, and white are still going to go 1 to 2 to 1 in each of these cases, and tall are going to go 3/4 to 1/4. So tall red is 3/4 times 1/4 is 3/16. Tall pink is 3/4 times 2/4 is 6/16. Tall times white is 3 times 1 is 3/16. All right. So notice that we got in here we got 9 that are tall and 3 that are never mind, we won't do that quite yet. All right. So we got 3/6/3. Well, I guess what I was going to say, this is 1 to 2 to 1 in terms of red, pink, white, because we got 3/6/3. That's the same as 1 2 1. Now short, we're going to take this 1/4 and multiply it by this, by this, and by this. So we'll get 1/4 times 1/4 is 1/16. 1/4 times 2/4 is 2/16. 1/4 times 1/4 is 1/16. So of the short ones, red, pink, white is still going 1 to 2 to 1. Of the tall ones, red, pink, white is going 1 to 2 to 1. But when we combine them together, we get this other thing. Then we get 6 plus 3 plus 3 is 12. 1 plus 2 plus 1 is 4. And that's a 3 to 1 ratio. So as far as tall to short, because all the ones on the top are tall, to short, that gives us 3 to 1 ratio, because it still does its thing by itself. Red, pink, white is 1 to 2 to 1. And when we multiply them together, we get this crazy ratio. Yeah, that's something you ever want to memorize. That's just something you want to be able to figure out. So let's say we take a curly fly, and we'll make this curly fly have purple eyes. Oh, that's a good one, because we could say curly purple. So we take curly purple cross to wild type. What are we going to get? Well, the purple will be all wild type, so it'll be all wild type for purple. But we're going to get 1/2 curly and 1/2 wild type. Right? How do I know that? Because this is, so we're using the curly from before. We already know it's a lethal allele. So we have to know that ahead of time or have enough data to figure it out. So now I want to do an F1 cross, but I want it to be a dihybrid cross. So this guy, so this wild type one has to be big C, no, it has to be little c for curly, and big P for purple. Because the big C by itself is lethal when it's homozygous. So this has to have 2 little c's, this has to have a big C and a little c. And purple is recessive, so that's going to be little p. So then this curly guy has to get a big C and a little c, and a big P and a little p. So he's going to be curly. He's going to curly because curly's dominant, and purple because purple's dominant, or wild type because wild type's dominant. So this guy's just curly. Just curly, and red wild type eyes. This one is little c because it's got wild type wings, and big P little p because it's got wild type eyes. But we want to do a dihybrid cross, so we want to make sure we are heterozygous for both. So this is a little VFL2 alert. So curly is one of those examples. So you'll see curlies like this, stubble, aristopedia. There's a bunch of these genes that are like this, where if we set up a cross, we get 1 to 1. We want to take the one that we know is heterozygous. So it's going to be that one. All right, so now what are we going to get? Let's think of each gene by itself. A curly monohybrid cross gives us 2/3 curly, 1/3 wild type. The purple one is going to give us regular 3/4 wild type, 1/4 purple. So what are we going to get overall? 6/16 curly. That's these guys. These aren't 16ths, these are 12ths. So 2/3 times 3/4 is 6/12, or 1/2. 2/3 times 1/4 is 2/12. Curly purple. 1/3 times 3/4 is 3/12. Wild type, so that's wild type for both things. And 1/12 just purple. So the infamous 6:2 to 3:1 ratio. Again, don't figure it out. Just figure out each gene by itself and multiply them together. Codominance is the same thing as incomplete dominance for the most part. The famous one is blood type. So if we got AB blood cross to AB blood, get 1 to 2 to 1. So that's our genotypes. Blood type would be type A, type AB, type B. 1 to 2 to 1 means it's a monohybrid cross. But what's happening is with the AA guys, so these are blood cells. The A gene makes some kind of an antigen. It's just some kind of molecule. It's actually a carbohydrate on the surface. So if you have an A allele, you get this. So we'll say A equals this kind of carbohydrate. Say B equals this kind of carbohydrate. So if you've got a B allele, you get the green one. So if you get the A and the B allele, you get both, and that comes up as type AB. If you've got just the B allele, you get the green carbohydrate cell surface molecule, and if you get just the A allele, you get just the red. So 1 to 2 to 1, that means it's a monohybrid cross. The fact that this guy shows both phenotypes, so it's not intermediate between two phenotypes. It's not whatever color is intermediate between green and red. What's that, purple maybe? No, I don't know. I'm terrible with colors. So it shows both green and red, so it's codominance. So there's another locus. So because A and B are codominant, neither one of them is dominant to the other, we get this sort of deal here where our A allele is given this designation, our B allele is given this. So the I is the locus, and the letter up here is the allele. And then there's another allele called IO, which is also sometimes called just little I. And so these are multiple possible alleles that can be at the locus. there's your locus. And you could have the I, it's the I locus, and you can either have the big A there, you could have the B, or you could have the little I. But any individual only gets two of these. So you can be big A and big B. You can be big B and I. You could be big A and little I. So any combination of those things is fine. And then, but these guys are codominant. So we can write it like this. They equal each other. Just with a regular allele, let's say that we had sepia. Wild type for sepia is dominant over mutant for sepia. So that we would write that as dominant. So these guys are codominant, so they just get an equal sign, but they're both dominant to little I. So if you have IA, I, you're type A. If you have IB, little I, you're type B. If you have little I, you're type O. So to put it all together, these are your different types of genotypes. Those give you A, this gives you O, and then if you get this, it gives you AB. So knowing that, you can do all kinds of crosses. Let's say you have, in a human, let's say we've got type AB blood and cystic fibrosis, which is a recessive mutation. And then somebody else has type O blood and is normal for cystic fibrosis. They have a kid. Let's say they have, so they can have this kind of kid. They can have this kind of kid. Alright, let's say these kids have a baby. Probably not the best genetics example in the world. Then what could you get? You can get big A with big B. Get big A with I. You can get big B with little I. And you can get, so I think that's all you can get. Alright, sometimes it's worth doing a Punnett square. Just keep ourselves honest. So for the A locus, we can get big A or I, or big B or I. So we can get these things. So these are 1 to 1 to 1 ratio. 3/4 big C, 1/4 little c. These are all 1/4. So we can multiply these probabilities to see what we get out of this. So in this case, we're saying this is normal. This is cystic fibrosis. We say, okay, well, what's the probability of getting cystic fibrosis and having type AB blood? Well, that's 1/4 and 1/4. You get a 1/16 probability of that. Not the best example. But the main thing is that you figure out whatever ratios are going on with one gene, whatever ratios going on with the other genes, and then multiply them together as long as there's independent assortment going on. So this we have independent assortment. We also have multiple alleles because we can have more than 2 alleles at a locus. And then we also have codominance going on. So there's lots of stuff going on with that particular cross. Alright, this is supposed to be quick, so I will okay, we did combining probabilities. Sex linkage. Oh boy, we better do this one real quick. Well, sex linkage isn't good to be done quick, so we're going to stop now, and this video's